fragment, but we do that so that we can match it up with the light reactions. If you do the reaction for this fragment, the free energy change is plus 477 kilojoules per mole, so that's unfavorable. We need 8 photons, and we need to couple this unfavorable reaction to this favorable reaction up here, and that takes 8 photons, and so we can calculate the energy of 8 photons from a formula that I gave you previously, and that energy is a negative 1594 kilojoules. And so you can see there's more enough energy in those 8 photons to carry out this reaction. Now, if you want to talk about the whole reaction here, that's going to require 6 times what's given down here. And so that would be 6 times 8, or 48 photons are required for the entire reaction to form one glucose molecule. So a lot of photons are required there. So that's just kind of the overall reaction kind of balanced out to show you where things are coming from. Now, the Kelvin cycle is an amazing cycle. It incorporates many of the reactions we've already seen in gluconeogenesis and glycolysis. It brings in a few key new reactions that we're going to talk about today. And it was all worked out by Professor Melvin Calvin, and he used one of the first to use radioactive carbon-14 to work this out. I've emphasized the use of radio to trace metabolic pathways, and this is one of the first and really most amazing uses of that technique to work out the entire Calvin cycle, which is named in his honor. And we're going to look at different parts of it. The whole thing looks kind of imposing when you see it all on one. At parts of it, I think we can get through. Is ribose 5-phosphate, and that is a simple carbohydrate.
A 5-carbon carbohydrate. Things we need to keep track of the Calvin cycle is the number of carbons. We're always going to be interchanging numbers of carbons as we go through this Calvin cycle. We start out with 5 carbons, ribulose 5-phosphate. We activate that with an ATP, ribulose 1,5-phosphate. We now have a phosphate on either end, and again it's 5 carbons. This right here is the critical reaction. What Calvin did originally is he radiolabeled carbon dioxide with C14, and then he put it in the chloroplast of a plant, and then he isolated all of the compounds that incorporated and how rapidly they appeared after he added the CO2. And the first molecule that appeared was 3-phosphoglycerate. The carbon dioxide that he originally added is C14CO2. And so the 3-phosphoglycerate from that critical experiment identified is the first compound formed during the Calvin cycle. So note we've got 5 carbons in ribulose 1,5-bisphosphate. We add carbon dioxide and we get molecule of glycerate, each containing 3 carbons. So that's 5 plus 1 is 6, and that's 2 times 3 is 6. And so we incorporated our carbons. And this is really critical because this is where we incorporate CO2 into a carbohydrate. The 3-phosphoglycerate is a carbohydrate. It's a small one, and of course you'll recognize it as one of the intermediates in the glycolysis pathway. Now, the rest of these reactions on this diagram here are just gluconeogenesis. You've seen all of these before. So we take glycerate, we add ATP to make 1,3-bisphosphoglycerate. We then reduce it, and this is where that NADPH comes in. That makes our glyceraldehyde 3-phosphate.
We then continue up the gluconeogenesis pathway to fructose 6-phosphate, and then we can take that on up to glucose itself. So that's the route that we get up to glucose. But then we have to regenerate the ribulose 5-phosphate, and that involves some additional complications that we'll see a little bit later. So the basic plan is we take 5 carbons, ribulose 1,5-bisphosphate, add a 6th carbon as CO2, make the 3-phosphoglycerate, that is the reaction, NADPH, and so forth, to regenerate the glucose molecule. So this shows us this
process. The fixation is stage 1, that's the stage. The reduction to fructose 6-phosphate at second stage, treat this ribulose 5-phosphate, third stage. This enzyme that carries out the key step here is called ribulose 1,5-phosphate carboxylase. And we simply abbreviate that as Rubisco. So I'll just be using the abbreviation here, Rubisco, quite a bit here. So ribulose 1,5-bisphosphate carboxylase, and so that's what we'll be calling it. Let's look then at the mechanism of this enzyme. By the way, this is the most plentiful enzyme on the face of the earth. This wins out over all other possible enzymes. There's just an enormous amount of this in all the plants and trees and everything else. So an enormous amount of this Rubisco is present in the biosphere. So the active site of this enzyme has a magnesium ion that is critical for the reaction. It also has an unusual lysine that has a CO2 attached to it that is ligated to that magnesium. That's kind of an unusual little thing. It helps to stabilize that magnesium. The magnesium is also held in place by other amino acids in the active site, including aspartate and glutamate. But what happens here is the ribulose 1,5-bisphosphate comes in and binds to the active site at the magnesium ion. And you can see that the ketone group in the ribulose 1,5-bisphosphate ligands to the magnesium, and then the OH on carbon ligands to it. And that particular electronic environment causes this key reaction here where we remove the proton, that proton right there, that goes away. The electrons on that bond become a double bond. It's called the ene dialate intermediate. So we have 2 OH groups, or 2 oxygens, that's the dialate function, and the ene is the double bond. So that is a critical stage in that mechanism. We then bring the CO2 in, and that also binds to that magnesium ion, and it is activated by the binding of the magnesium ion. The electrons in this double bond are then used to attack the carbon of the CO2, and we've now formed this critical linkage between that carbon of what used to be ribulose 1,5-bisphosphate and the CO2. So that step right there is the critical fixation step in which CO2 is incorporated into a carbohydrate. And that's what's responsible for the production of all of the carbohydrate on the planet now and in the distant past. And that, of course, went on to form oil and gas and coal by geological processes. So this series of steps is the most abundant on the planet, the enzyme is, and it's one of the most important in producing food and energy and everything else we need for life. Now, this is the same thing, but shown in a somewhat more diagrammatic fashion here. We have, this is the ribulose 1,5-bisphosphate, the phosphates on each end. There's the ketone at position 2, 2 OHs. A typical
carbohydrate with 5 carbons. We then, as we showed in the previous slide, form the double bond between carbons 2 and 3, and now we have an O- on carbon 2 and an OH on carbon 3. We then react with the CO2 with the electrons in the double bond, as we saw before. The CO2 now is attached to carbon 2, and at the same time this OH on carbon 3 now becomes a ketone. Then in the next step, water attacks that third carbon, and we now have that water down. And then we have the breaking apart of this. This carbohydrate right here now contains 6 carbons. 1, 2,
3, 4, 5, and then the CO2 is the 6th. We now break that bond between that and that carbon. The top part ends up here as the 3-carbon fragment. Initially it's a carbanion, but a proton comes in and forms 1 molecule of 3-phosphoglycerate. The bottom part here comes out as just plain old 3-phosphoglycerate. So these are both the 3-phosphoglycerates. We get 2 of these. Again, the critical thing is to count up carbons. That's 5 carbons. The CO2 is the 6th. We've got 6 carbons here, 6 carbons here. We break that critical bond right there, and we get 2 molecules of 3-phosphoglycerate. I think you can see from the coloring here that the CO2 is incorporated into this carbon 3 of the 3-phosphoglycerate. Only one of these contains the CO2. That fragment comes from the lower half of the original molecule. So that is how we get the carbon dioxide into carbohydrates, appearing first as the 3-phosphoglycerate. So that's the key reaction. That is the essential reaction of the Calvin cycle. Now, the rest of this is just going to be regeneration. And this is the whole thing. This looks like kind of a mess, but I think this is a much more informative picture than the one in textbook. They managed in this book to get everything on one sheet of paper and coded, so you see what's happening to all of the carbons. A little hard to see in this classroom. I would recommend that you print this off in color if possible for one of your fact sheets on the next exam. And let's just go through this. I have more detailed slides a little bit later, but we start the ribulose 1,5-bisphosphate. That's what we had on the previous slide. Due to that, we 2 kills of phosphoglycerate, as you can see there. And then we go on to the next step here. And the next step is just part of gluconeogenesis. We use energy in the form of ATP to make 1,3-bisphosphoglycerate. So that's just part of gluconeogenesis. We reduce that to glyceraldehyde 3-phosphate. We then equilibrate that with the dihydroxyacetone phosphate. We take one of those molecules, one of those, and we make fructose phosphate. Remove the phosphate to get fructose 6-phosphate. One of these then goes off to form glucose. So that's where we're done as far as making glucose. All of the other reactions that are on here are used to regenerate this ribulose 1,5-bisphosphate. And again, keep track of the numbers of carbons. So that's 5. Those are 2 molecules of 3 each. 3 carbons, 3 carbons, 3 carbons. Put those 2 together, we have 6 carbons, 6 carbons. We then have an interesting reaction here, which is called a transketolase. I'll show you that in more detail in just a minute. But what we do there is that we combine together in different ways this fructose 6-phosphate to make a 4-carbon molecule called erythrose 4-phosphate. That then is combined with
a 3-carbon dihydroxyacetone phosphate to make 7 carbons, cellulose 1,7-bisphosphate. One of the phosphates is removed. We then react the 7 carbons with 3 carbons, and we make 2 molecules each containing 5 carbons. So 3 plus 7 is 10. We make 5 plus 5 is 10. Just make sure all your numbers of carbons end up. These two are then converted to ribulose 5-phosphate and phosphorylated to the ribulose 1,5-bisphosphate. We're done, okay? This diagram, you need to look at it a little while. The number of arrows indicates how many times that reaction has to occur in the full balanced Calvin cycle. So it's a little hard to count them here, but those are 6 arrows, so that reaction occurs 6 times. And you can see that reaction occurs once, and that reaction occurs twice, and so you just count up the number of arrows to get how frequently that reaction occurs. Notice what you need for making this reaction work. You need ATP there and there. You need reducing power in the form of NADPH right in that position there. So that is where you need these key inputs of energy and reducing power for the Calvin cycle to work. Then all of these reactions here, this kind of go along without any further input of energy or reducing power. So that's the overall Calvin cycle. If you look at this in your computer and you print it out in color, you'll see where the radioactivity goes. That red there on the CO2 comes from the red CO2 is colored red here. And as you can see, that ends up in the middle 2 carbons of the fructose 1,6-bisphosphate. And it appears in different ways in all of these other types of molecules. So try to follow that on this chart. Okay, now I'm just going through some of these reactions a little more detail so you can see them here. This is one of the first of these reactions. You combine the fructose 6-phosphate, 6 carbons, with glyceraldehyde 3-phosphate, 3 carbons. You make erythrose 4-phosphate, 4 carbons, and xylulose 5-phosphate, which contains 5 carbons. So 5 and 4 is 9, 6 and 3 is 9. You've not lost or gained any carbons. That's a good deal. We call this a transketolase reaction. And that's because a ketone reacts with an aldehyde. So the ketone means transketolase. It makes a new aldehyde and a new ketone. So that general class of reactions is called a transketolase. And you can look from the color coding here what we've done. We've taken the top 2 carbons of fructose, stacked them on all 3 carbons of the glyceraldehyde 3-phosphate to make this xylulose. So those 2 came from the top carbons. We're left with the bottom 4 carbons for this erythrose 4-phosphate. So that's the transketolase reaction, and we have several other examples of that in this Calvin cycle. Here then is a aldolase reaction, the same type of reaction we saw in the aldolase reaction in glycolysis. We take 4 carbons, erythrose 4-phosphate, 3 carbons of dihydroxyacetone phosphate. We make 7 carbons. 4 and 3 is 7. This is an aldolase reaction because we react an aldehyde with, as you can see here, an alcohol. That's the part of the molecule that reacts. So an alcohol with an aldehyde is called an aldolase reaction. We make that new bond between this part of the molecule and that aldehyde reaction right there. So that's the same type that we saw in gluconeogenesis and glycolysis, but with different molecules here. But 4 carbons, 3 carbons make 7 carbons. We then hydrolyze off the phosphate to make the cellulose 7-phosphate. The next step here is another transketolase reaction. Here we take 7 carbons, the cellulose we just had there. glyceraldehyde 3-phosphate, and we're reacting a ketone with aldehyde to make a new aldehyde and a new ketone. And basically what we are doing here is we're taking the top 2 carbons, stacking them on top of the glyceraldehyde 3-phosphate to make the xylulose 5-phosphate, and then we've got the remaining 5 parts down here is the ribose 5-phosphate. Again, count the carbons. 7 and 3 is 10. 5 and 5 is also 10. So we've gained no carbons and we've lost no carbons. Again, a transketolase reaction. Okay, one more step and we're done. This here is a reaction in which we simply isomerize the ribose 5-phosphate to the ribulose 5-phosphate. We've seen this type of isomerization before in glycolysis. We started out with an aldehyde. We reduced that to an alcohol and oxidized that alcohol to a ketone. So that's all we've done there. We've isomerized the ribose to ribulose 5-phosphate. The xylulose 5-phosphate is the same molecule as the ribulose 5-phosphate, but the stereochemistry at that carbon is different. In the Fischer projection formula, that OH is going to the left. Here it goes to the right. We call that an epimerase reaction. So now we've got our ribulose 5-phosphate. Now all we need to do is use another molecule of ATP and form the phosphate group on that number 1 carbon, and now we have our ribulose 1,5-bisphosphate. So that completes then all the reactions of that cycle. And let me go back to the overall picture, which maybe makes a little bit more sense now. The key reaction is the ribulose 1,5-bisphosphate reacts with CO2 and then is split apart into the 2 molecules of 3-phosphoglycerate, as you can see there. Okay? And then we use ATP to add another phosphate to make 1,3-bisphosphoglycerate, reduce it to glyceraldehyde 3-phosphate, and then undergo gluconeogenesis. This whole thing is just standard gluconeogenesis. Then we can take one of these molecules, make glucose, and the remaining ones we have to regenerate our ribulose 1,5-bisphosphate, and we do that with those transketolase and aldolase reactions that I've just talked about. We take the 5-carbon molecule here and combine that to make the erythrose 4-phosphate, and the cellulose, that's the erythrose, and that one there, etc. So all of these reactions in here are the ones that I showed in more detail on the other slides. So it's kind of complicated to look at, but when you've got these kind of charts, I think it's fairly straightforward and logical. But again, given the complexity, it is really an amazing achievement that Kelvin was able to work out all of these intermediates from this C14 radioactive tracer experiment. Okay, any questions on this Kelvin cycle? So that's the so-called dark reaction. We've used the light energy to produce ATP and NADPH and oxygen, of course. Then we've used in the Kelvin cycle that ATP and the NADPH to produce glucose. And that's where all the carbohydrate on the planet comes from. Now, the only other thing I'd like to talk about here is regulation. And it turns out you don't want to have these enzymes active all the time. That would create lots of problems. And so they're under very tight regulation in the plant. And this just kind of indicates what's going on in the chloroplast under different conditions. In the dark, at night, we are not producing any NADPH. So the NADP is all in the oxidized NADP plus form. We're not reducing ferredoxin, which remember is an intermediate in the production of NADPH. So that's in the oxidized form. We're not pumping protons, so the proton concentration is a bit higher out in the stroma. By the way, the yellow is the stroma, the part that's inside the chloroplast but outside the thylakoid lumen. And then the gray part here is the thylakoid lumen, the inside of the thylakoid. So we don't have a high concentration of protons there because we're not pumping protons, and we do have magnesium inside the thylakoid lumen in the dark. Then we turn on the light, the sun rises, all of these light reactions begin to occur. We produce NADPH, that becomes reduced. We reduce the ferredoxin, that becomes reduced. The magnesium is pumped from the inside of the lumen of the thylakoid to the outside into the stroma, and the protons are pumped from the stroma into the lumen. So all of these things change when the light is turned on. And all of these factors go towards activating the Calvin cycle. So we want to turn off the enzymes of the Calvin cycle during the dark. We don't need them and they would be wasteful. And we want to turn them on during the light. And one way we turn them on is that when this ferredoxin becomes reduced, it acts to activate several of the enzymes of the Calvin cycle. And one of these is shown here, this is sort of the general pathway. The light reduces the ferredoxin to the reduced form of ferredoxin. That then reduces another molecule called thioredoxin, and it normally in the dark has a disulfide bond there, but reduced ferredoxin can reduce the disulfide bond to 2 sulfhydryl groups. Then the thioredoxin in turn can reduce one of the inactive enzymes, which has a disulfide group, into the active enzyme, which has the 2 sulfhydryl groups. So these enzymes involved in the Calvin cycle are generally inactive when they're oxidized with a disulfide and active when they have the 2 sulfhydryl groups. And then slowly in the dark, if you turn the light off again, oxygen will reoxidize these disulfides, these sulfhydryl groups back to the disulfide. So that would return it to its state in the dark. So again, the sequences of events is that the light reduces the ferredoxin. The ferredoxin reduces thioredoxin to the sulfhydryl form. The reduced thioredoxin reduces a number of enzymes to the sulfhydryl active form, and then we carry out the Calvin cycle. Now, the enzymes that are activated in this fashion are shown on this slide, and this again is all in plants. And rubisco, this main enzyme that carries out the very first reaction of the Calvin cycle, that's one of the main ones that is activated by thioredoxin. So in the light, the thioredoxin reduces a disulfide bond in rubisco and activates it, and that carries out that key carbon fixation step in the Calvin pathway. It also activates the fructose 1,6-bisphosphatase, which is involved in the gluconeogenesis step of the Calvin cycle. It activates the glyceraldehyde 3-phosphate dehydrogenase, the sediheptulose 1,7-bisphosphatase, a number of other enzymes involved in other pathways, and also this ribulose 5-phosphate kinase that's involved in the Calvin. So this thioredoxin is a key enzyme in regulating the activity of a number of these plant enzymes involved in the Calvin cycle. So that's a very important pathway. Let me just back up. I just wanted to show the overall, yeah, this is good here. Just to put this in picture from what we talked about last period, this is the overall reaction of the light reaction of photosynthesis. So we excite the photosystem 2 with light that oxidizes the water to oxygen and produces quinol. The quinol transfers the electrons to the B6F complex, and we have a Q cycle that pumps protons across the membrane. The electrons are then passed on to plastocyanin and to photosystem 1, where another photon of light boosts the electron up to reduce the ferredoxin. That reduces NADPH using this enzyme here. The proton pumping here then is used to drive the synthesis of ATP. So all of this is what's occurring during the light reaction that we talked about last period. And then if we and then these are the summary equations that I just talked about. And then this is the entire set of dark reactions of the Kelvin cycle, where we're using this NADPH and the ATP to produce glucose. Okay, so there's the activation process. Now, there is a problem. There's always a problem. Rubisco is an amazing enzyme, but it has a defect in it, and that defect is illustrated right here. Remember that we start out with a ribulose 1,5-bisphosphate. The enzyme is then converted to this enediolate intermediate here, and this is where the CO2 binds. But we don't always have very large concentrations of CO2, and so a certain percentage of time, oxygen can get in there and react instead of carbon dioxide. And even though this enzyme is amazing, it hasn't devised a way to prevent all of the oxygen from reacting. It doesn't occur terribly frequently, but it occurs frequently enough to cause a problem. When the oxygen reacts, it binds to this here and forms what is called here a hydroperoxide intermediate with a peroxide OO bond there. Then water reacts, and we release a phosphoglycolate molecule. The top half is the phosphoglycolate. The bottom half that's released is the 3-phosphoglycerate. That's just the normal compound that's released. But this phosphoglycolate is an undesired side product. And so even though this enzyme is amazing, it still can't get away without having a certain amount of oxygen reacting to form this phosphoglycolate. And the enzymes, the plants have developed this amazing pathway to regenerate ordinary carbohydrate out of the phosphoglycolate, but it takes energy to do that. So they've taken this into account, this defect in the Rubisco. They've taken that into account by another enzyme I won't go into to process this phosphoglycolate, but it reduces the efficiency. And all of the plants on Earth that are of the C3 type, maybe you've heard of C3 plants, but they're like the ordinary plants and trees that we have. All the trees are C3 plants, and most of the plants are. They all have this defect. This process is called photorespiration. Just respiration means reaction with oxygen, and photo means in the presence of light. And so this photorespiration process here limits the efficiency of photosynthesis in these C3 plants. Well, another class of plants has found a way to minimize this, and these are the C4 plants that include corn primarily and related plants. And you might know that corn is one of the most efficient crops on the planet, and that's because it is a C4 plant. And the way it does this is it has two types of cells. The outer cell in the leaf is called the mesophyll cell. The inner cell is called the bundle sheath cell, and that's where the photosynthesis occurs. This pathway involves enzymes we've all seen before. We take phosphoenolpyruvate, an intermediate in glycolysis. We carboxylate that to oxaloacetate. We've seen that reaction before. We reduce it to malate. That's part of the citric acid cycle. Then the malate is transferred from the mesophyll cell to the bundle sheath cell. It is oxidized to pyruvate, and CO2 is released. That CO2 enters the Kelvin cycle. The pyruvate is then converted back into phosphoenol pyruvate by an enzyme we haven't seen before that uses ATP and phosphate to make AMP and pyrophosphate. So in one step, we go from pyruvate to phosphoenol pyruvate. Okay, the net reaction here is simply a way of increasing the concentration of CO2 in the bundle sheath cell. So this, if we have a low concentration of CO2, this little cycle here increases that concentration of CO2. The more CO2 you have, the better you can compete with the Rubisco and have that react with CO2 rather than oxygen. So this minimizes photorespiration. So the C4 plants like corn are more efficient because they have this extra little cycle that boosts the local concentration of CO2 and minimizes that unfavorable photorespiration reaction. You'll notice that this does take some energy to do this. We do take NADPH, but we gain it back here. But it does take ATP, 2 equivalents of ATP, as a matter of fact, because we use that twice. So we have to pay a price to do this, but it's not as bad as that photorespiration side reaction. So and there's a lot of work being done currently trying to incorporate this type of pathway into plants other than corn by genetic manipulations. So this C4 pathway is, this is called the C4 pathway, by the way, so that's a very interesting feature of these plants. Okay, so that's all I have. Are there any questions on this photosynthesis? If not, we'll take a short break and then we'll begin working some problems. Okay?